When did they
No, yes.
and do this?
Yes, now. Now, move it. They will stand this. Thank you.
This is a device to move the background. It's okay. You moving?
It's here. You carry.
Okay.
We are so excited to have you all here. Today, we will be introducing the first wave of our local investigator funded by Hevolution, and each of them will share with us what we have been working on and what lies next until they finish their projects. So pretty much they are giving us the progress research that we are doing over a year. Also, next, we will have some exciting announcement at 3 o'clock, so stay tuned, and it's going to be in the main arena. Let's hear from our investigator. Next, Dr. Anas Abdulrahim. Please join me to welcome Dr. Anas. Dr. Anas Abdulrahim is a clinical scientist at King Faisal Specialist Hospital and Research Center. He is associate professor at Al Faisal University College of Medicine with more than 100 peer-reviewed manuscripts. Dr. Abdulrahman research interest focus on biomarker discovery using integrated multi-omics, mainly metabolomics. Go ahead, Dr. Thank you.
Thank you, Dr. Ali, and thank you for Hevolution for inviting me to this event. I would love to share our one-year research since we had the first grant. I will be talking today about metabolomic revolution, unlocking the secrets of disease-free aging. Yeah. First of all, I will share with you some background about aging and aging hallmarks. It's basically the biology of aging associated with genomic instability, telomere shortening, epigenetic changes or epigenetic clock, mitochondrial dysfunction, cellular senescence, and altered intercellular communication within the same tissue. Aging is a significant risk factor for many diseases, including the cardiovascular disease, neurodegenerative, and many others. The study of aging, it is a multidisciplinary study where cover multiple aspects of biology, computational, and molecular biology. The primary objective is to comprehend a fundamental mechanism of aging is still at the age it still needs to be investigated and need to be understood nicely. The reversible aspect of aging is very crucial, where we could, in metabolic level associated with these aspects, can be reversed and can be changed drastically, and you could maintain the health and the age, the biological age of a tissue or organ. Looking for inside our cells, if we look inside the cells, we have multiple layers of biomolecules from genomics, proteomics, and metabolomics. Genomics is just four letters of very small molecules. Proteomics is around 20 letters, while metabolomics is very complicated, it's more than 100,000 molecule. So the complexity of chemistry increases as we go from genome to metabolome. However, the biological complexity is getting narrower, so metabolomics is very complicated, less complicated from biological aspects. But nicely, we would love to, nicely that also the metabolomics is the most insensitive biomolecule to the environmental and physiological changes. This would make metabolomics to provide more functional readout about our genome, that it is embedded in our body, and it helps also to predict the protein abundance associated with the protein abundance as well, and the environmental influences that might trigger some other expressions and genomic profile inside our body. Metabolomics, it's any small molecules in our body, including the molecules with molecular size below 1500 Dalton. That covers all the vitamins, sugar, peptides, drugs, drug metabolites, food additives. This includes also the microbiome that it is secreted inside our gut, outside our body for many functional aspects. If we care about metabolomics, our small molecules, it's very important if we are looking for diagnostic clinical assay. So if you walk in any laboratory, you will find more than 95% of the laboratory assays based on small molecules. 89% of the known drugs in any pharmacy is based on small molecules. 50% of those drugs were discovered based on pre-existing small molecule in our body. So 30% of the identified genetic disease associated with inborn error of metabolic disease. So if you are looking for biomarker discovery or drug discovery or gene function, so metabolomics will be a guidance, or it will be the best tool or the ideal tool to work in during our research. So the future depends on what we do today. And the Hevolution funded project, we started with 423 healthy individual patient sample recruited for this particular project. The age range was between 18 and 79 years old. We run them in our platform. We call it mass spectrometry, untargeted mass spectrometry platform that detect around 12,000 ion between positive and negative moods. And after filtration, data cleaning and filtration, we ended up with 6,630 metabolite. So this is the workflow that we run our samples on. We get samples from consented people. We extract the metabolites and we run them in our platform and mass spectrometry to get data that we are trying to understand and make sense of out of these data. So this is the aging profile based on grouping, and we group them in from A to E based on 1 decade difference between both of these cohort, and we're trying to have equivalent match gender matching and in every subgroup. But within our group, we've also recruited a chronic patient cohort that has, some of them they are obese, the other are diabetic, some of them they have blood hypertension, and some of them they are having more than one disease. 21 of those has like 3 of them. What we try to understand at this stage of the project, we are trying to find a panel of metabolites that associated with age. And we found out that we have 20 of those metabolites that are extremely screaming significantly increases or decreases while we age. And those metabolites, we tried to evaluate them from biomarker standpoints, and we found the area under the curve of rock cave analysis, the area under the curve for 2 or 3 metabolites, it could reach up to 100% prediction based on carnitine, indole pyruvate, or 5-hydroxysterone sulfate. Those molecules usually make sense to be declined as, for example, dihydroesterone sulfate. It makes sense to decrease while we are aging. And the area under the curve was 80% by its own, but when you combine it with a panel, and we always looking for panels of metabolites that improve the accuracy of a prediction. And the carnitine was one of the metabolites that increases. This is another example while we are grouping, and this is associated with mitochondrial dysfunction as we age. And those are the list of 20 metabolites. Around 6 of them still unknown in terms of annotation. So when you look in the database, it's still challenging and they are not there. So the red ones, they were down-regulated, the green ones were up-regulated, and the ones with the star, those are endogenous metabolites inside our body. And those metabolites, they have 2 classes, endogenous or exogenous. Exogenous, that means we get them from outside, environmentally, or from food, or from our medications. The endogenous from our biological mechanisms that we have in our body. So I will go with just a simple example. If phosphoinositol, this is kind of lipid molecules. It is associated with dysregulated lipid remodeling, such as acetyltransferases or phospholipidase. Or it might be associated also with reduced lipid turnover inside age cells. It is lipid pre-oxidation or membrane damage, also associated with elevation of these molecules. And finally, dysregulated phosphoinositol signaling, PL3K and AKT signaling. So this is the whole study. I consider it is a hypothesis generating study. So next we are going to validate this finding through additional experiment, such as the proteomics, tenaces profile that we have already started, and it's part of our objective, and lipidomics is also part of our objective, and finally the transcriptomics and the proteomics experiment to conclude the function and to validate this marker at bigger cohort, and additionally in using another experiment. In conclusion, the panel of metabolites, it was discovered in this cross-sectional study and disease-free cohort, and it needs to be extrapolated also to disease models. These metabolites were classified in endogenous and exogenous, and they are associated with biochemical pathways in our body, or to trigger aging-related genes in our based on the external metabolites. These metabolites would serve in healthy a digital twin, and is a background monitoring for disease such as the chronic disease, diabetic, non-diabetic, or the obesity. For sure, this panel needs to be extensively validated to be used and utilized for further investigation. Thank you very much. And I have a poster over there for another findings in this study.
Thank you. Next, Dr. Marwa Adruish. Please join me to welcome Dr. Marwa. Yes, okay. Dr. Marwa Adruish is Associate Professor of Molecular Biology and Microbiology at King Saud bin Abdulaziz University for Health Science in Riyadh. Her research focuses on establishing sustainable strategies to investigate the gut microbiome role in aging, aiming to promote healthy aging and enhance quality of life in Saudi Arabia. The floor is yours.
Thank you. It's working? Okay. Thank you, Dr. Ali, for introducing me. So I would like, first of all, to thank Hevolution for inviting me to deliver an oral presentation about the progress of my research. Last year, I presented briefly an idea about my research, and today I'm glad to happy to present the progress so far. So as you can see, the title of my presentation is Towards Sustainable Research on the Gut Microbiome and Aging in Saudi Arabia. So I would like to start my factors that are common with aging are basically influencing our gut microbiome. So we as a scientist, can we intervene to revert the action of these environmental factors? The answer is yes, through prediction of unhealthy aging using AI models, using of probiotics and prebiotics, personalized diet, and we can estimate the biological age and predict and promote the healthy aging. So I was curious to know in the literature about the studies already published in the gut microbiome and aging space. On the left-hand side, you can see the publications of gut microbiome and aging in PubMed. Using only PubMed database, there is a growing number of publications in the aging and microbiome. I was also interested to know if there are ongoing clinical trials regarding the gut microbiome and aging, and the answer yes. In total, there are 32 clinical trials already registered in clinical trials database, and the leading country is the United States, most commonly using dietary supplement as an intervention to modify the gut microbiome. However, the clinical trials here are basically focused on aging-associated diseases, mainly neurodegenerative disorders. So what about healthy aging? This is an ongoing systematic review we are conducting at the moment in order to characterize the gut microbiome composition and function in healthy aging in the world. So these are the results. Can you see the distribution of countries that are published studies in healthy aging? And getting an example, one of the blue zones in the world, Sardinia, Italy, in which individuals living until to 100 years with healthy aging. However, the data from this systematic review indicates a lack of studies, not only in Saudi Arabia, but also in the Middle East region. So we, as a group of scientists from Saudi Arabia, we got the initiative from Hevolution, with Hevolution support, to start establishing gut microbiome and aging research in Saudi Arabia. So our aim is to understand healthy aging through the lens of gut microbiome, and specifically to perform metagenomic examinations of fecal specimens collected from healthy Saudi individuals across different age groups. Why is this important? Because in Saudi Arabia, we completely have different genetics, different diet, and different lifestyles, and all these factors influence the gut microbiome composition and function, and ultimately influence the aging. So what are other methods? We are following the analysis workflow for microbiome in order to determine causality and enable therapeutic development. So as you can see here, our research project focusing on stage 1 and stage 2 to do the metagenomics and then data-driven hypothesis generation. In subsequent projects, we will be able to validate the hypothesis using animal models and in vitro models, then to move forward with clinical trials and intervention. So this is the pipeline of our study. We basically collect samples from healthy Saudi individuals. The clinical samples we are collecting is stool samples. The stool sample is processed, DNA, microbial genomic DNA is extracted, and then we are preparing the sample for next generation sequencing using two approach, targeted metagenomic sequencing and shotgun metagenomic sequencing, in order to have a comprehensive information about the microorganisms and their functions in the gut microbiome and the study participants, along with the metadata collection, including demographics, social data, lifestyle data, diet data as well. So we basically collected samples from Riyadh city, okay, from Saudi individuals, from different places, from employee clinic at King Abdulaziz Medical City in the National Guard, and from Riyadh community as well. We excluded individuals with intestinal disorders and individuals who use antimicrobials because these influencing the gut microbiome negatively. Also, since our study is focusing on healthy aging, we are excluding participants with chronic disease, with acute diseases, with cancer, neurodegenerative diseases as well. So the results so far, we have 217 healthy Saudi individuals eligible to participate in the study. They provided their stool samples, they provided comprehensive metadata information. And we divided the study population according to different age groups. Here is an example of some of the variables collected from the participants. Most importantly, diet is a huge determinant of the gut microbiome composition and function. And we in Saudi Arabia have completely different diet habits. So we take the initiative also and to pioneer this field to study the diet and gut microbiome in the context of aging in Saudi Arabia. So this slide shows the diet data analysis pipeline. We collected the diet data, comprehensive diet data using validated food frequency questionnaire through in-person interviews. The raw data is processed using MyFood24 nutritional analysis software, and then the produced data is analyzed using SPSS statistics software. So we'd like to show some of the preliminary diet data analysis. So here is the protein consumption and carbohydrate consumption. As you can see from the figures, protein is very important to the gut microbiome composition and function, and carbohydrate as well. So in terms of saturated fat and total fat, these are the results, the preliminary results of fat consumption at different age groups. And fiber as well, because fiber is very important for our gut microbiome composition and function. We are interested to investigate and analyze the fiber intake consumption in both genders at different age groups in Saudi Arabia as well. Last but not least, there is an interesting article published in Nature, studies the influence and the impact of caffeine consumption on the gut microbiome. So this study was conducted in population from United States and United Kingdom. So we took the initiative to study also this variable in the Saudi Arabian population, given the popularity of caffeine in the Saudi Arabian population. So the study started in February 2024 and will be completed at the beginning of 2026. We are at the moment doing extraction of microbial genomic DNA, preparation, the metagenomic sequencing, and development, the bioinformatic pipeline. And then we will adopt AI and machine learning modeling in order to predict the healthy aging in the Saudi Arabian population. I would like to thank Hevolution for supporting our project. I would like also to thank leadership at our institution, the Science and Technology Unit, and also the research team, my colleagues who are presenting here and supporting me. Thank you so much.
Thank you, Marwa. Amazing. So next, we will have Dr. Majid Al-Hamrani. Dr. Majid is Associate Professor of Physiology at Taif University. He got his PhD from the University of Monash in Australia. His research focuses on liver studies and liver aging. Go ahead.
Thank you. First, I would like to thank the organizer for giving me this opportunity to present my work, focus on the aging liver. So I will start with some background and previous work. Transcription factors are master regulatory for cell identity that control the activity of aging, of gene and cell function. But how does their activity change in aging, and are these activity change randomly? We address these questions as part of international collaboration leading by Nifzikar Lab from Australia and published recently in Cell Metabolism, and uncover a conservative mechanism across cell type linked to organism maturation process. We found that rising activity of AB1 transcription factor progressively reprogram the transcriptome of tissue and organ toward adult tissue function during maturation, while shutting down the development process. This process is hijacked in aging by ongoing AB1-driven chromatin opening via stress and inflammation, and which further dams the activity of youthful gene, which we believe underpin many aging phenotype. So mechanistically, the activity of youthful gene is shut down via siphoning of AB1 cofactor from early life to youthful gene regulatory element toward AB1 exposed chromatin in combination with mid-down regulation of cell identity transcription factor expression. So rather than a normal process, the transcription factor networking and expression is affected by rising activity of AB1 and hijacking of development process. We studied the chromatin remodeling of more than 45 cells during maturation and aging. We also analyze, as part of our study in cell metabolism, we also observe similar remodeling pattern in hepatocyte during maturation and aging. We found that in motif enrichment, the region that are open during hepatocyte maturation transaction and aging transaction have high enrichment of AB1, which on the other hand, the region that are closed during maturation and aging have high enrichment for transcription factor for hepatocyte cell identity. It's important to understand the aging in liver, because the liver is the causing of different type of disease. With age, there is increased risk of variety of disease, including the metabolic disorder like the measles. And in Saudi population, there is about 30% who are affected from that. And by 2030, it will increase to 50%. The hepatocyte is a structure organ. It consists of repeating hexagonal called the liver loop. At the corner of the liver loop, the portal vein, the blood flow from the portal vein toward the central vein through the synovial vessels. The hepatocytes, as we can see, have different color. They are arranged according to the cell growth. According to their position, they have different rules. For instance, the pericentral hepatocyte, they play an important role in glycolysis, lipogenesis, glutamine synthesis, while the hepatocyte that's surrounding the periportal play an important role in cholesterol biosynthesized protein secretion. This difference in their function and their alignment is controlled by cell signaling, in particular the Wnt and the RAS signaling. And it's been shown recently that the EGFR playing an important role in RAS activation. So, to address how age-related chromatin remodeling manifests the tissue level, we have two aims. The first one is to perform a special transcriptomic data generation and analyze for mouse liver. The second one is as part of ongoing experiment. We will assess the change in protein level and test gene modulation on cell models. The special transcriptomic is a new cutting-edge technology. It allows us to study the change in the whole transcriptomic tissue and intact tissue. The vision of a special transcriptomic profile at 55 micromil resolution, which is not single cell, but it's still not far from it. So in our experiment, we had 3 different animal groups. And number is 5. We have the young mice, which are almost 2 months, the middle-aged 10 months, and the aged mice, which are between 22 to 24 months. We cull the mice, collect the liver, sectioning, embedding, perform the special transcriptomic sequencing, and then mapped back the reading on the image. We mapped the special dots along the pericentral to periportal axis using different technique, and one of them was the special landmark. As we expected in young mouse, we found that the dots that surrounding the pericentral express the gene that associated the pericentral, while the dots that surrounding the periportal express the gene that link with the periportal. However, from middle age onward, there was no change. Not in the pericentral have no effect, while in the periportal there was a diminish in the arrangement. So to assess that, we global map the change in receptor ligand cofactor. As I mentioned in the beginning of my talk, the cell signaling, especially the Wnt and the and the RAS signaling, play an important role in arranging the liver. So we assess the change in we assess the global change in receptor ligand cofactor in the neighbors dots of our special transcriptomic data based on their expression. We found number of the receptor ligand, receptor cofactor interaction that have an aging change, and the one that score high and occur the most earlier was decline in interaction with EGFR, as you can see in the left, right, and the right, on your right top side. In line with that, there was a significant reduction in the expression of the EGFR from the middle age onward. To link this with our chromatin study, we assessed the gene regulatory element of we found that the EGFR regulatory element of the gene regulatory element of EGFR lose accessibility in aging. Aligned with our previous finding, there was no change in the pericentral gene. There was also no effect with the Wnt signaling, since the Wnt signaling play an important role on that. So, to summarize, we established a high-quality spatial transcriptomic data set. We mapped the global change in receptor ligand interaction. We identify loss of periportal zonation signature in aging, which was associated with the reduced RAS signaling. The activity of youthful gene like EGFR appears to be dampened in response to AB1-mediated chromatin modeling, aligned with our siphon model, which we published in Cell Metabolism recently. And this change may underpin age-related liver disease risk. Next, we will assess the change using protein validation. Also, we will do some in vitro studies. Finally, I would like to thank all my colleagues from Taif University Dr. Wala, Dr. Abdul Hakim, Mr. Ahmed, Ibtisam, Iman, Nujud. Also thanks to our international collaboration, Dr. Christian, who came all the way from Australia, and his team, King Saud University, Professor Khalid Al-Swat, and finally, Dr. Hamza Habib. But during the question, I will be happy to answer any questions. However, if anyone, thank you. If anyone have any question regarding the cell metabolism, the corresponding author, Dr. Christian, he came all the way from Australia. This is him. Thank you.
Thank you, Majid. So next, Prof. Valerio Orlando. Prof. Orlando earned his PhD from University of La Sapienza in Italy and completed his postdoc in molecular epigenetic at the University of Heidelberg, where he developed the chromatin immunoprecipitation technology. He started his independent career in the San Rafael Institution in Milan and led a project, a first flagship project with a 30 million euro budget. In 2013, he moved to KAUST in Jeddah and established epigenetic program. Go ahead.
Thank you very much. Thank you, particularly to Hevolution for having supported this project. So what I'm going to talk about is the so-called dark side of the genome. So that half of the genome is composed basically by repetitive elements, and these repetitive elements, their function in evolution has been very well studied. However, the RNA component that comes out of them remains largely unexplored. This is going to be the topic of my talk. Now, these RNA, actually, the expression of these part of the genome is actually quite intriguingly connected to two fundamental aspects of cell physiology and tissue homeostasis, which is epigenome plasticity and also innate immune response. Two elements that in aging become problematic in the case of erosion of the nuclear architecture and in the case of chronic inflammation. That is the two hallmarks of aging. So these are the topic we're going to solve. And this also connects with an important element of how we measure aging, biological aging, which is the frailty. So the capacity, as was discussed this morning by Felipe Sierra, is how resilience matters for understanding aging. So in this context, retrotransposed RNA seem to be at the crucial point, at the crossroad, and we call them like resilient RNA, as they are sensors of what comes from the environment. And what is actually been shown is that in fact there is a deregulation of repetitive elements of transcription during aging, and in particular in the latest phases of life. So this was been driving our study, and we published two stories that are the kind of yin-yang aspects of it. In the first story, we followed overexpression of line elements as a characteristic of aging, and devised a technology or an approach where we can actually knock down the line elements in that context and rejuvenate or basically block progeria progression and so on. In the other case is following the expression of line elements in the context of cell differentiation, and in this case in particular bone differentiation, and discovered that osteoporotic cells or mesenchymal stem cells from osteoporotic patients do not express properly line elements at the proper time. And in that case, we provided these cells with line elements RNA, and this restored the ability to produce bone in a very complex manner. I don't have time to go into that. So in order to have a clue of how the regulation, the global regulation and subcellular localization and so on of these transcripts changes with time, we took an omics approach, multi-omics approach, and in particular we're going to talk about the nuclear compartments and cytoplazmic compartments. We combine transcriptomics, we combine proteomics, metabolomics, and also technologies that allow us to map where these RNAs operate in the chromatin and which components they actually target. So the material is human primary fibroblasts. We have, sorry. age spanning from 1 to 96 years old. It's a pretty robust type of code. So in the case of transcriptome, and we use a sub-fractionation, so we can fractionate cells from cytoplasm to nucleoplasm to bona fide chromatin and resulting fractions from there, and then do RNA-seq and follow the profile of the different types of RNA. We then, by looking at transcriptome, I love the previous talk. What we see is a clear transition here. There's a precise time of our life when things change. Change in the sense that cells change identity. So the point here is what is aging in terms of changes in cell identity, the program. So these cells are not sick. They simply do something else, and at the spatial transcriptome level, they probably differentiate in different cell types that we don't know. We have to characterize them and so closely and so on. So there's a clear transition time. It is a nonlinear profile of the same, basically expressing the same concept. If you look at the proteome, the proteomics of the same subfractionation in the different manner, don't have time to go into the details of the technology, but we observe exactly the same thing. So this is consistent with the phenotype. Now talking about heterochromatin components, they always shift, but they shift at a precise time, and surprisingly, the major components of heterochromatin decline quantitatively, in movement. In another set of data, metabolic enzymes that are associated with chromatin do not. So what it changes here is probably the targeting here. Now, if you look at repeats RNA, this happens to be the most important transcript that actually change in this context, and the line 1 in particular. But again, the transition happens at the precise mid stage of life, not later. So in later stages, the change in transcription is not very prominent, whereas in a particular set of time, it does matter. And we can now narrow down specific subfamilies that participate in the process, which is pretty important in the TECOM. So in conclusions, we see a clear crisis time at the middle age, as we know, it happens, that clearly transition to something else. And this nicely corresponds to when retrotransposons become deregulated in sense. Now, the point is whether retrotransposon RNA in this context, thinking of the connection with chromatin and innate immune response and chronic inflammation, are they drivers or passengers of the process? As an epigeneticist, I have no problem saying that transcription factors actually command. No problem. So we have to understand what is the connection between transcription factors and the rest. So who is there? Now, to partially address the question, we use a method we devised some years ago that allows us to map the RNA in vivo, like the chip technology now is RNA specific, and we hope then to see to generate the picture. That's where we stand, basically, with the study. We hope to complete it in the next couple of months. Thank you very much for your attention, and to my great team for having participating in this adventure.
Thank you, Valerio. Thank you. Great. So our next speaker, Dr. Talili Barhoumi. Dr. Talili is a research scientist and team leader in the Medical Core Facility and Research Platform at KMARC. He obtained his PhD in Human Movement Science from University of McGill. Dr. Talili research interests focus on the implication of adaptive and innate immunity in hypertension and cardiovascular diseases. Go ahead. Okay.
Ready, madam. Okay. Thank you, Dr. Ali, for this presentation. So our work is about the role of adaptive transfer of T regulatory cells in aging-associated cardiovascular diseases. So aging is usually associated with dysfunction in immune system. a decline of immune responses in elderly people. This dysfunction dysregulation is associated with both innate and adaptive immune system and leads to low-grade inflammation or inflammation, or it's called inflammaging. When this inflammation reaches the vasculature, usually it leads to cardiovascular diseases. One of the key regulator of immune tolerance and also inflammation is T regulatory cells. T regulatory cells are usually characterized by dysfunction in elderly people, regardless of their number. And even the balance between T regulatory and T effector cells and function, usually it's dysregulated in elderly people. Previously, we shown that T regulatory cells are associated with vascular damage and cardiovascular diseases, and also that sick regulatory T cells are usually lose function regardless of their number in the host. So later on and more recently, we shown also that adoptive transfer of T regulatory cells or fresh, boosting the body with fresh T regulatory cells, it prevents vascular inflammation, oxidative stress, and cardiovascular diseases. So for this, we hypothesize that maybe adoptive transfer of T regulatory cells may may prevent vascular damage associated with aging. For this matter, we isolate T regulatory cells from young mice and T regulatory cells from old mice, and then we inject them to old mice to see the effect on cardiovascular damage. Mainly, we try to see the effect on heart, kidney, and vasculature. So T regulatory cells are responsible and modulator of both intestinal inflammation and systemic inflammation. The measure by inhibiting directly or indirectly the pro-inflammatory or metabolite, pro-inflammatory cytokine in systemic or pro-inflammatory chemokines. We choose these three parameters to start our study. So you know that TMAO, which is generated from TMA, is responsible. Recently, they shown many studies. They shown that it's responsible for cardiovascular disease, and also it's implicated in cardiovascular disease associated with aging. This metabolite is stimulated and directly by pro-inflammatory cytokines, and also the T regulatory cells may inhibit the release of this cytokine. So we try to see in our model if there is an effect of young T regulatory cells or old T regulatory cells on TMAO release. So we found that when we inject old mice with old T regulatory cells, we see a slight decrease in TMAO release. More interestingly, when we inject them with young T regulatory cells, we see a significant decrease in TMAO release with a big number in our model. Also, on the other hand, we know that CX3CR1, which is one chemokine, that it's a pro-inflammatory chemokine, what is highly expressed in immune cells, implicated in inflammation. And also, on the other hand, it's regulated by T regulatory cells. We don't know if maybe there is an effect in aging associated with inflammaging. So we check the expression of this chemokine in heart of mouse. We see that there is a slight increase with all Treg cells injected to the mice, and this effect was blunted when we inject them with young T regulatory cells. In summary of this preliminary data that we found that adoptive transfer of young T regulatory cells may have an effect in intestinal inflammation and also systemic inflammation that could be through the TMO release that we found it significantly downregulated when we treat them with young T regulatory cells, and maybe through the axis CX3CL1, CX3CR1 axis. So all these data need more validation, and for our next work for this project, we will validate our results, and also we will see in other organs, especially kidney and spleen, to see the pro-inflammatory cytokine with innate and adaptive immune system. I want to thank you all, our team, our research team, and also Science and Technology Unit in KMARC. Thank you for you all.
Thank you all for coming, and thank you for our speakers, and hope to see you next year at the same place with more researchers and scientists. So now we will conclude. We will not have time for Q&A session, but because we have another announcement in the main arena starting now, so we would like to go there, and thank you for coming and sticking around.
Hazi's talking right.
All right.
Hazi's
Okay.
talking right behind us, so I think that's more interesting. Are you giving a science talk?
So okay. All right. So good afternoon. My name is Ali. I'm the program officer at Hevolution. Um, and welcome to the session, the Global Talent Pipeline in Saudi and Beyond. So before we get started, let's begin with just a brief introduction from each of you. So if you can go ahead and just introduce yourself briefly.
Sure.
Yeah.
Well, thank you for having me, Ali. My name is Laura Niedernhofer, and I'm the director of an Institute on Biology of Aging and Metabolism at the University of Minnesota, about the opposite kind of weather from what you have here.
Okay, yeah.
I am also a professor of biochemistry, molecular biology, and biophysics, and I work largely from a perspective of DNA damage as a driver of aging, and then cellular senescence as a therapeutic target, and I use a lot of mouse genetics.
Oh, nice.
That's my model system.
Oh, okay. All right. Go ahead.
Yes, so my name is Andrew Dillon. I'm a professor at UC Berkeley, also a Howard Hughes Medical Institute investigator. And I've been in the aging field now for almost 30 years. And I primarily have been trying to figure out, why young cells are so resistant to stress, whereas old cells are not.
Perfect.
And that's left it very open for me to move around in different areas of science. And now I've really focused on how cells are responding to pathogenic attacks as a major driver for aging, and trying to figure out that biology. And I don't direct anything.
Nice.
I try to direct my lab as best I can, but
Perfect.
that's hard to do.
Perfect. Excellent. Thank you. To keep the field of health span moving, we need to attract and retain talent. However, the field itself is facing significant challenge, as we know that 20% of highly skilled professional and around 30% of early career in academia, they're moving, trying to find better opportunities. In addition to this, 1% of global workforce involved in the aging and health span research and development. And in addition to this, the health span field is a nascent field, which add more layer of difficulty and make the job is really hard to attract talent. So to tackle this, we need a diverse strategies to build or to make a global network to attract talent, governmental action, partnership with academia and universities, collaboration with the private sectors. So from your perspective, what does it take to build a talent ecosystem, similar to the one, for example, in the US? Is it purely rely on salaries, or deeper infrastructure need to be taken? You can start, Laura.
Okay, well,
Yeah.
this is a great point. That's an interesting statistic that only 1% of scientists work
Yes,
in the aging
I
field.
was
I
surprised.
would personally argue that all of us work in the aging field, because aging affects so many processes, right?
I think it's just based on who directly work on that
fair
field.
enough,
Yeah.
fair enough. So what would it take? I mean, certainly I think broader education to young people, even starting in elementary school, to really understand how our world is going to change if we don't find a new way to address our aging population. And so I think education, awareness is going to be incredibly helpful. We've even had opportunities to make commercials for football games because they're so popular.
Yeah.
So just something to just get the word out there and share the message with everybody. I don't think you need any unique infrastructure.
Okay.
You need investment, there's no question. That's always the primary thing for attracting talent. But then I think the other thing is just also raising awareness that if you want to have a huge impact with your career, particularly in the health industry, this is the way to go. This is going to have greater impact if we can find a way to target aging biology and translate it into humans, much greater impact than treating or curing a single disease.
Awesome. Andy?
Yeah, I mean, those are really, really great points, Laura. I think also, layering on top of that is that aging can also be viewed as very diffuse. There can be a lot of aspects of it, and I think that a young person coming into the field may get very distracted early on, is that, they have the opportunity to work on degenerative diseases, cancer, everything is affected by aging. And so it becomes very hard to focus. And I think that if we were better at, making the message that you can work on cancer and aging, aging is a major driver. You can work on aging as the driver for this disease, that disease, this process, that we could actually focus students a little bit more closely and get them funneled into working on aging. I think there's also a pushback from those other fields, cancer, for example, and you can probably speak a lot more about this, about aging scientists coming into, aging researchers coming into it, saying, I want to apply the rules that we have for aging onto cancer. And they don't want to hear that because they want to focus just on the cancer. They don't want to focus on, what is the leading cause of it. They want to focus actually on the disease. So I think our messaging and getting more sort of focused on the questions that aging can address, I think would definitely create a larger pool of people wanting to go into it.
So, but is there like a specific, I would say like move or initiative should be taken to build like complete ecosystem, like for countries, for example, to attract talent? Like you mentioned, like education, for example, from the early stage, to tell them about the field in general, or research, or so. Is there like anything else can come together with the education to push the field and build a complete ecosystem?
Ooh, I think Andy raised a really good point. We see that trainees very often are going to enter biomedical science or clinical practice because of a family member that had Alzheimer's or cancer, and so they want to make a difference. And I think raising awareness that you might be able to have a greater difference, make a greater impact if you study aging along with your interest in neuroscience or with cancer. Because let's face it, we don't have cures for all cancers, we don't have cures for all Alzheimer's, despite incredible investment and decades of time. So one example I can give you that I think sadly failed in the United States was when I was starting my career, there was an effort to have funding at the aging and cancer interface. And you could easily do this with aging and neuroscience. But just getting an education in both arenas is really, really important for actually understanding the biology behind it. So I think trying to merge disciplines and offering very high-tech training, because right now we need to rely on single-cell evaluation, spatial analysis of tissues. So I think the combination of awareness, some funding at the interface of a couple of disciplines to bring in different perspectives, broad training, and then also some real high technology and bioinformatics.
Excellent, perfect. Andy, if you have any...
Yeah, I don't have much more to add on that. I mean, I look at my trainees and the ones that go on to biomedical research, in the companies, it's like you said, it's like they're wed to a disease because of a family history or something. So that's what's driving them. I mean, you do get the curious student that really is curious about aging. And those are 1 out of 10 that I get in my lab. The others are driven by a disease process. And I think that having aging partnered to disease process would definitely drive more people into the field.
Okay, excellent. And training, educations, like say story inspiring. Do you do you think the global partnership and international collaboration between either like in the level of governments or in the level of educations will kind of promote these things? And what kind of a program do you think it will help to kind of push the field forward in attracting talents?
Yep.
So you can go ahead.
Well, I can speak to, so locally what
Yeah.
we have done
Yeah.
is we've taken advantage of two mechanisms of NIH funding. So this
Okay.
is specifically from the National Institute on Aging.
Nice.
And they have a program that's called an R25,
Mm-hmm.
and that is for undergraduate students. And what it does is it provides them an opportunity to go to a great lab in aging research, and they have to spend 10 weeks there in the summer.
Nice.
And they really get so involved in that they end up trying to come back
Oh.
and stay in touch.
That's really amazing.
It's a super mechanism.
Yeah.
And then there's another one that's called an R33 grant mechanism. And that's a little bit different because it's really meant to create educational tools.
Okay.
So you can do it in a video format or a certification in geroscience, for example. And those are very valuable because what's really tricky is to create new graduate programs or create new areas
Sure.
of clinical training. That just takes a lot of work because there's a lot of regulation.
Yeah.
I think these ancillary things that create education, whether it's at the wet bench or theoretical, I think those are very valuable. I would add that the collaboration, in my view, is probably the most important thing that can happen. I personally went to a European lab for my training, and that was life-changing for
Wow,
me.
yeah.
It's absolutely essential for creating a network of scientists that you can always lean on and count on for supporting your career, and it changes your science dramatically. So one thing we have done as an example is we created an alliance that involves now 8 different universities across Europe and the United States. And we get together for an annual meeting. We share students and trainees. And it's just become very powerful. And I think our next step is going to be a clinical
Wow, that's,
translational research program.
that's
yeah.
really amazing. That's really cool. Andy, if you have anything to add.
Yeah, I mean, if you think about it, if you could somehow organize, there are certain hubs across the world that are very focused on aging, where there's critical mass of people that are sharing ideas locally. So, and for example, where I come from, the Bay Area, we have Berkeley, UCSF, Stanford, and the Buck Institute. In San Diego, they have the Salk Institute, UCSD, Burnham, and Minnesota, they have a large focus there. New York, there's focus. Boston, there's focus. Even in, if you look at Cologne in Germany, this is only 10 or 12 years old.
Wow.
The Max Planck there, along with the university, a partner to make it probably one of the best aging centers there is. And so if you could somehow tie those together, get those centers to work with each other and then work amongst each other, sharing students, doing these exchange programs. I mean, we have an R25 as well, where we bring in students for 10 weeks to Berkeley to do the same thing. But wouldn't it be great if those students working at Berkeley also interacted with the students at Stanford, also interacted with the students from Minnesota, got this, camaraderie going on with each other, where they knew each other across the planet, all working on aging. So there's some way to link those together. I tried to do this. I proposed this to HHMI, and they kind of said, no, we're not going to do that. Because I don't know what resources would be required to do it, and the organization actually do it. How vast it would be, but it would be fascinating if you could actually get the trainees talking to each other. I think that's where innovation is going to drive. I mean, me and Laura can talk, for days and come up with ideas, but then we get on a plane and we go back and we forget to tell the people in the lab about it.
Yeah.
Because we get busy. But when the students talk, then things happen. The students and postdocs. So if we can figure out some way to get these nodes, the trainees there working and talking with each other, I think that would be transformative.
Maybe Hevolution should adapt such a program
Well,
Please do.
to do this.
as long as you provide the administrators
Yeah,
as well,
yes.
because I think it's the getting it to
They're
all
gonna- yes.
the work is going to be
Yeah.
fascinating.
And talking about like life changing and inspiring when you if you do like in a different lab. It's same thing happened to me personally when I got a Fulbright and went to City of Hope to do a 6-month research. It's completely changed my views. It changed like being like before I was a student, I wasn't like kind of have a broader view, but getting into the research field and then going like for 6 months in a different lab, pretty much completely new area. So it's just kind of like ignite like every single thing like in your brain, trying to expand and really make you like more into what you're doing. So
Yeah, I think the way you could do it is that you have the students go to each one of these centers, and then after the 10 weeks, you have one symposium where they all come together.
Yes.
You know, you can do Zoom calls and everything while that 10 weeks is happening, but then you bring them all together at the end, and everyone meets and they exchange and they create. Because that's really what drives fields, is that like you become friends with the people there,
Yes.
and
Yeah.
you grow a field from talking and collaborating, and and you only collaborate with the people you like.
Yeah.
So
True.
having something early on in your career focused this way could really make things go a long ways.
that's really great.
And I think you could actually do it in two directions, right? So if you had trainees in different labs and then brought them together after their experience, that's one way to do it. But the other thing that's been really successful is like the marine biology laboratory course, or just NIA summer training program. So these are all educational tools where everybody comes together first. They don't know each other at all. They're bringing very diverse science, and then they're trying to figure out how to write a grant or what's next in their career development, and then they go back to their own space. But they've already formed a club that's going to support each other, and that's really important as well.
Awesome, great. So sometimes it's easy to attract talent, but the issue sometimes is how to retain them in the field, which is like I guess more challenging. So, in your opinion, how do we inspire like young talent that who get attracted to the field to stay and not to leave? So is there like any specific strategies or from your experience just to keep them like as long as possible in the field, to push the field forward and promote like innovation in the field?
Do you want to tackle that
Yeah,
first?
I'll tackle that from my own personal experience.
Okay.
So I think in my lab now, 18 of my trainees have gone on to start their own labs. And, when I go to meetings in the field, like a lot of my former trainees are there, and what happened to them is that early on, they got in the field and the other people in the field were very supportive of them and really mentored them. I mentored them as well, but the field mentored them and brought them into the field. They weren't critical. They weren't, it wasn't competitive. They were very supportive. And so my people got to grow up in this field, and then they started their labs, and instead of moving to other fields, they stayed in it because of what was being supportive. And so now it's great. I go to these meetings, and they're doing it, they're paying it back. So they were mentored and nurtured when they were young, and now I see them doing it to the next generation. And I think that that's super important for a field, because I've seen fields, chew themselves apart. Cancer has done this, cancer fields have done this, apoptosis field, where it's so competitive and there's no collaboration and there's competition and no one is helping each other. And so why would you go into a field like that if you're a young person? So you want to go into a supportive field, and I think the aging research community is right now one of the most supportive fields there is. And so if we can continue sort of that ethos, there's no funding for that. It's just people behaving nice,
Yes.
behaving as they should in science.
Attitude sometimes is very
Attitude
good.
is entirely it, is that you have to, mentor and see that there's a whole new crop of people coming that have new ideas, they have new technologies, and help them develop it. I think as more senior in the field now, it wasn't so long ago we were the junior people, but now more senior in the field, we have to realize that and really support the more junior scientists.
Excellent. Perfect.
Yeah, I think that's really an excellent point, and I've been very mindful about picking fields that are kind to each other and they don't eat their young, as they say. And for me, DNA repair field was like that when I got started, whereas telomeres was very contentious, and I was terrified of transcription factor field for the same reason. But I can promise you the aging field is very kind right now.
Awesome.
Yes.
And there's such an urge to mentor and help if you just ask.
Yeah.
And so I think it's a really good choice. The other thing I started thinking about that might be a solution is, well, let me back up and say this. Mid-career is really tough. I mean, it's really tough in the United States. So one thing that's really good in the US is we give a lot of people an early start. You could get a grant with a little bit worse score, and if you're young, you will get that grant. If you're older, you just won't.
Yeah.
So mid-career is tough. But I was thinking if there were a way to maybe make like TED Talks or podcasts that really show what you can get out of a career when you're at a stage like Andy or myself, I think that would help retain people. I mean, there's so many things you get to do. I mean, you're hanging out with smart people all day long, and you're getting to see junior people blossom, and you can travel all over the world. And just to highlight the happy parts, because everybody thinks, oh God, I'm not going to write grants for my entire life. But it's a great career. It's just fabulous.
So you're saying it's kind of like a side job beside like doing a podcast or going out, talk to people, just like to inspire other and keep yourself in the field.
Right. I just think there needs to be more communication to make it clear how great it is to be a scientist.
Yeah.
And to stick with it, even though there's hard times. Yeah, grant writing can be painful.
That's right.
But there's all these other pluses that come with it. So.
That's really amazing. Cool. With any field, I guess there is always a barrier for that one. So do you think like there are like most like or let me say, what is the most critical barrier in the Healthspan field? Is it the field is still nascent and naive, let's say, somehow compared to others, or the funding? It's not like really accessible to everyone, and it's still not much. So what is the barrier like in the field that you see like will stop like attracting talent?
I think I don't mean to talk over you. So my lab, we work in model organisms that are rapid, but we also do mouse work and we also do stem cell work. And with students coming in, I never give them a mouse project because I'm like, you have the time to do one, maybe two experiments, and if they're wrong, your PhD is over with. And that's just a resource limited problem. If we could create something for these, the problem with aging research is it's long. The experiments are long. And for students to stay and wait for a result, it's torture. It's where there are neighboring labs that do biochemistry, they get a result in a week.
Yes.
In our lab, we're lucky if a result happens in 2 or 3 months.
True.
And so this is something, if we could somehow create some mechanism where we can do more experiments with these trainees so they could have more feedback quicker, I think that would be, and it's not changing model organisms, it's not changing organisms that's going to do it. It's changing the way that we're doing it, not doing just one mouse experiment at a time, but doing 10 at a time, for example. I think that would be a game changer for the younger scientists, something like that.
So a couple of ways to fix that really is longer grants. That would certainly help, particularly in the field of aging, so that you had stable funding and you could really do 2 mouse experiments, say back to back. That would take 6 years.
Yes.
It just does. The other thing you can do to fix that is to have infrastructure that provides the resources for these experiments so you don't have to wait for animals to age. And so the NIA has done a great job of that, and we're able to just get animals for free that are already 2.5 years old.
Yeah.
And that really jump starts experiments. I mean, you could get a graduate student to start right there
Yep.
and repeat the
Yeah, they don't
experiment.
need to wait for a couple of years.
Yeah, there was something when I was at the Salk Institute. We did something similar where we had a grant from the Glenn Foundation for aging, and we devoted, a certain amount of that money to have age colonies of mice that were specific to Salk researchers.
Yep.
Nice.
So genotypes that were specific to that institution, so that anyone could go in,
Oh,
and it was transformative. It allowed people to immediately pick up and do something quickly. If we could do that on a center-wide basis, I think that'd be phenomenal.
Yep.
Because it's the genotype that you need
Yep.
that's aged.
Yep.
Yes,
Exactly.
and,
yeah.
and you never know when you're going to need that aged genotype. So
right.
it's always kind of frustrating.
Yep.
This is really excellent. So just to final comment about what we need to keep the global pipeline remain sustained. So what is the things that we need to adopt in the field to keep it sustained for next generation?
Yeah. I mean, honestly, it always comes down to funding.
Okay.
It just does. And
Okay.
so right now, I think you have an incredible opportunity locally here. And I would really hope that the best and the brightest really take advantage of this situation.
Yeah.
But funding tends to go like this. So
Sure.
that's why collaboration and networks are so important too. So maybe all of a sudden the federal government in the United States will give a ton of money to aging. And then if you already have a network, then it's easy to share those funds and that experience and that wealth. And so I think those are the two most important things in my view: funding and collaboration and networking. Really.
That's very cool. Cool.
It's an input-output thing. So we've talked a lot about getting people into the field, but people go in the field because they look up and they say, where am I going to actually go after this? And, many of them are like, oh yeah, I'm going to go and I'm going to run an aging research lab. But some of them are like, no, I actually want to go into industry. I want to go into biotech. And so, 15 years ago, there were no aging biotech companies. 10 years ago, there wasn't really any. But now there are some biotech companies that are showing up. So now students are looking and they're saying, now I get my PhD. Now I can actually go work at Calico or Altos or some of the other biotech companies. And I think you need to have a bigger culture of that. Is that if you have a bigger community of aging biotech companies, then more people will go into aging research to work in those companies. And I mean, we were talking about what's going to happen in Saudi Arabia here, in your city up north, about making a biotech center. If you make that a hub for aging research, people will come because they see that there's opportunities, and that makes them excited. And we see that in the Bay Area as well.
Excellent. So as we wrap up, final questions. If you could change one policy to enhance talent acquisition and improve retention, what it be and why?
One policy.
Yes.
I didn't know there was policies that were inhibiting this.
Well,
What do you mean? Yeah.
It's like I wouldn't. Okay, let's not call it policy, but
Okay.
one thing we talk about mentorship,
Yeah.
funding, and education, networking, collaboration, but one thing and why you think it's the most important one.
Well, I came up with an idea, it just popped in my head.
Okay.
One is the regulation of education drives me crazy.
Okay.
So it's very hard for me to start a brand new graduate program, like a master's degree or even a new course based on biology of aging. It's very hard to get accreditation and final degree.
And things like
Yeah.
that.
And then I would say it's even worse in the clinical arena. I think every medical student should be educated in geroscience, aging physiology, and to get, at least in America, the regulatory body that monitors clinical education and residency programs and internships, it's next to impossible to change it.
Wow.
And that needs to be altered because this is a really rapidly changing
Okay.
world, and this
Perfect.
is good. This is really important biology.
Nice.
Yeah, no, I think that's great. My own selfish is like, I definitely want to increase the pipeline of students and postdocs, right? That's my own selfish because I want to see more.
Okay.
But that's not going to solve the bigger issue. I think the bigger issue is you got to invest more at the top, create more jobs at the PhD level for people trained in aging biology. So that means, more professorships, more biotech companies, more institutions that are focused on aging research, so that these people have a place to go to find jobs. And I think that if that opens up, then the bottom begins to come in faster. So my policy would be, let's invest at the top and create more jobs for PhDs in aging research.
Excellent. Changing education and more postdoc. And
Not
PhD.
postdoc, post-postdoc.
Okay,
Yeah.
postdoc. All right.
Post-postdoc.
We still have maybe 1 minute if you guys have any question. So. Okay.
So one of the things that Texas has done separately from California when they're importing a lot of talent from that is no taxes. And when you have affordability, headquarters of companies are moving, to your point, creates more jobs, feeds the whole bottom up into Texas as a building force and building an ecosystem, which is extremely mature in San Francisco. So, by the way, thank you so much for an amazing talk. I just wanted to say that.
Well, so could I comment on that? I mean, you're bringing up, excuse me, an excellent point. We might have a huge shift from the East Coast, West Coast doing science because of climate change and
Taxes.
forest fires and taxes and hurricanes. And so, yeah, I'm very happily nested in the middle of the country, just like yourself.
Yeah. And the cost of graduate students. I mean, it's enormous on the coast. Yeah. Yeah. I think it's going to completely change.
Yep.
All right.
Thank you. It was very nice talk. It was enjoying for me. I am working in a research center, and one of the challenging that we have is sometimes we have a lot of students or postdoc that need to have a training in our lab. And this is good to have much people, but one of the issues is the losing a lot of resource for training them. And sometimes you don't have enough time just for mentoring them, and you need sometimes to give them for another person or the trainee or one of the technologists to take care of them. What's your opinion to save this challenge in the research center?
Yeah, so I run my lab very hands-off, and I actually partner people into little pods where you have a mentor inside the lab. So as a student will be mentored by a postdoc, but normally I have a student will have 1 or 2 postdocs mentoring them, so there's no falling through the cracks situation. And so it's the way I organize it so that not all of it falls on me. And also, not all the decisions are based on me, because I make irrational decisions a lot of times, and the students follow them for 6 months, and it's the wrong choice.
Yeah.
So as long as there's a committee involved in the decisions and the mentoring, I think it goes, it speaks volumes for the student's success. The other point is, the postdocs and the students that do the mentoring, they're testing out their water to see if they can actually do this. And so it's a good way to go through that. I don't know if that answered your question, but that's sort of the approach that I take so that I don't lose people because of lack of mentoring.
Well, I guess one approach that we have taken is that we create like a summer course that's a wet lab experience that's for a whole group of students at once, to get everybody exactly at the same point where they're now skilled. They have a toolbox of molecular biology skills that they can take to almost any discipline, which all relates back to aging anyway. So that's one way to do it. The other thing I thought about is something maybe for Hevolution, and that is I actually get an applicant to my lab every day of the week, and it breaks my heart when I have to turn somebody down. Yeah. But maybe keeping an inventory of mentors, even if they're only available, by Zoom or, not in person, but mentors or labs that have openings but don't have the pool of talent. I just feel like if there were more strategic ways to place people, we could maybe fill that gap. Because it's terrible when somebody falls through the cracks, isn't it? It's just
Turning someone down, it's really bad. You feel like
it's horrible.
Crushing them.
Yes.
Yeah. All right. Thank you. Thank you for sharing your thoughts inside about talent retention and attraction in Healthspan, and thank you for for sticking around with us. Thank you.
Thank you very much.
Thank you.
Yeah.